In this tutorial, we shall deal with post-translational modification of protein. These can be found under biochemistry, and let's first go to post-translational modification of single amino acid. We see here a wide variety of modification, which is composed of maps which describe the mechanism and tables which compile all the proteins that have been shown to be modified in Plasmodium falciparum. Let's go first to a very familiar reaction. This is the phosphorylation of protein, either serine or threonine, by protein kinase and their subsequent dephosphorylation, or the phosphorylation of tyrosine by specific kinase and its dephosphorylation. We can now go to the phosphoproteome, and which compiles all the proteins which have been this is the table, which is compiled from 5 independent investigations, here color-coded. And in the table itself, we can see that each of the proteins is labeled with the colors of the different investigation which has been identified. Obviously, the more color you have, gives a higher reliability to this identification. On the other hand, in the case of spermidine synthase, you see that has been identified only in one investigation, likely in the NADP-specific glutamate dehydrogenase, and this casts some doubt about the validity of these identifications. We shall go now to another modification, here protein lysine methylation. You see that by successive activity of the metal transferase, we get monomethylated, dimethylated, and trimethylated lysine residues on a protein. On the other hand, the demethylation can equilibrate or control the levels of mono, D, and trimethylation of the protein. And here we have the table of confirmed methylated lysine residues on Plasmodium. You can see that we have here indication about monomethylation, dimethylation, and trimethylation, and the position of the lysine on the peptide where methylation did occur. We now go to the role played by chaperone in protein folding and structural modification. We see here that either the nascent polypeptide is going out of the ribosome and then it undergoes several interactions and with other compounds, the GRO-EL and GRO-ES, and here we see how different variation of those heat shock protein interact directly with the ribosome and hit the peptide as it comes out. Here we see that we have a table that compile all the gene coding for chaperone regulators, for location of the various chaperone in different infected RBC compartment. And here is an example of the HSP70 chaperone cycle, and here is the location and function of the different HSP70 infected erythrocyte. We see that different variants appear in the nucleus, in the cytosol, in the mitochondrion, in the apicoplast, in the parentophorus vacuole, and eventually in the cytosol of the infected cell. Now we should go to ubiquitin-dependent post-translational modification. We know that reversible modification of protein through the attachment of ubiquitin is an essential post-translational regulatory mechanism in eukaryotes. The conjugation of ubiquitin or ubiquitin-like protein has been demonstrated to play a role in growth, adaptation, and homeostasis in all eukaryotes. Here is an example of the role of different types of ubiquitins in the machinery of the ER dealing with misfolded proteins. You see that there are different ubiquitin, UBC7, UBC6, and so on, which participate in the different regulation. And this is an example of the role of ubiquitin in the cell cycle by promoting the anaphase. And these are the details of the chemical reaction taking place during modification of ubiquitin, starting with the E1, E2, E3, and so on. Probably the best known ubiquitin-related process in cells is a proteolysis of ubiquitinated protein by the proteasome system. Here we see all the participants, the ubiquitin activating enzyme, conjugating enzyme, and the ubiquitin ligase, the movement of the ubiquitinated proteins into the proteasome, which is made up of the 20S proteasome, the lead subunit, the base subunits, and the peptide comes in to go out as peptide and release ubiquitin for recycling. And here is the table of all the protein which participate in the proteasome mediated system, all the subunits and their corresponding expression times, and from here we can get a deeper view into these intricate systems.